the invitation. And yes, so I'm a postdoc in Julian Donward's lab in Cancer Research UK that just became the part of the Francis Crick Institute. And we are having this project as a collaboration with Dr. Ali Tafazoli's lab in Southampton University, and we are actually interested to disrupt the interaction of two protein complexes. So I will kind of focus on these three proteins, which is RAS, B110 alpha, and RAS and CRAF proteins. And we're going to use cyclic peptides as a method to identify this inhibitor. I've only started on this project 6 months ago, so I will go through the main core of the background of the project and the technique and what we want to achieve and some preliminary results. So if we go through RAS proteins, so for people who maybe not so involved in the field, so RAS is one of the GTP, belongs to the GT-based superfamily of proteins. It consists of HRAS and RS and KRAS, and it's involved like any G proteins. It's involved in transducing cellular signals that arise from the cellular membrane. They're also involved in many cellular functions, crucial ones actually, which is like cell growth, proliferation, differentiation, and activation. And they also, the way they also start the activation, they interact with main target proteins that they generate or activate downstream signalings like PI3K kinase and RAF pathway. And they have been found that they have been mutated in almost 20% of cancers. And one of the most common mutations that we get to find in RAS proteins is glycine 12. And this is just a table to summarize where do you find the ratios and the percentages of RAS mutations in different cancer and different tissues. So if we also look at the probability and where the three RAS proteins, where do you find most of the mutations, you tend to find that G12 in KRAS is highly mutated, more than the glutamine 61, whereas in NRAS you find higher mutations, glutamine 61, and HRAS you find a kind of an equivalent amount of mutations in glycine 12 and 13 and glutamine 61. The sequence similarity between the proteins it's identical up to amino acid 85, and it starts to vary mostly at the end, which is the phenolization domain, where it kind of keeps them on the active state on the cellular membrane. So RAS protein is also, obviously, it's a switch protein. So when it interacts with it's inactive in a GDP state, and it's active in a GTP state. And there are two main switches that are actually quite important in this kind of shift or activation process, which is switch 1 and switch 2. Switch 1 here is just with the white, highlighted with the white color, and this the switch 2 is purple color. And you can see that there is a strong change in the loops where they are involved when they move from the inactive to the active state to try to interact with this gamma 3 phosphate of GTP. And there are 3 amino acids that are quite important, and that are involved in switch 1 and switch 2, that they start to move in to interact with this third phosphate, which is threonine 32 and tyrosine 32, threonine, and glycine 60. And you can see that there is strong movement on the interface around these 2 switches, but the rest of the protein is almost the same, doesn't changes under these interactions. So as I said before, it's one of the reviews that from my supervisor that RAS activates many cellular signals. We are interested in the 2 pathways of the RAF-ERK pathway and B110 AKT pathway. So PI3K is obviously it's a lipid kinase, and it phosphorylates PIP2 to PIP3. So it adds the extra third phosphate, and it's divided in 2 classes. Class 1A, it contains 3 proteins: B110 alpha, beta, and delta. And class B, class 1B, it contains B110 gamma, and all of them have the similar structure or the similar domain distribution. All of them have an AB domain, an RBD domain, which is we will intensively focus on this one, and a C2 and helical kinase domain. In our lab, we found that RAS interacts with all the BI3K proteins except B110 beta, and B110 alpha, that's been found that it's highly mutated in obviously in breast cancer and endometrial cancers, and then 80% of these mutations they found that it exists in the kinase domain, or sorry, in the helical domain and the kinase domain. And back in 2000, Backel have discovered certain mutations that they found in the RBD domain that when he performed this mutation, it eliminated the interaction between B110 gamma and RAS. So equivalently, our lab got interested in these mutations, and they used the equivalent mutations of B110 alpha, and they found that, you found these two mutations, these two amino acids, serine and lysine, that are reserved among all the species, and they found that these mutations made B110 highly resistant to oncogenic RAS proteins, and obviously this was consequently, they found that it had a strong effect in the reduction of tumor. So this is just images of mice that contained KRAS G12D mutation, and in the wild type you find there's the size of the tumors, whereas in mice that had the mutation where it had the two amino acids mutation that affected or inhibited the interaction with RAS, you found that there's strong reduction in mice. So I'm going to be interested in the structure, obviously, and the biochemistry and the biophysics behind the scene. So, but this is just the biological background. So if you look at the interaction between the RBD of B110 gamma and KRAS, again, you find that the switch one here, lighted in red, is strongly involved in this interaction with the interface with the RBD of B110 gamma. And what you find here, actually, I showed in yellow. These are the yellow ones are the interface amino acids that are involved in this interaction, and it interacts through mainly the interface is mainly through a beta sheet in the RBD domain on the N-terminus of the RBD domain. RAF is also a protein kinase. Instead, the BI3K was a lipid kinase. This is a protein kinase, but it's also been highly involved in mutations in cancer tissues in melanoma. But it also contains an RBD domain, and it interacts with RAS proteins, and oncogenic RAS have a strong effect on the constant activation of RAF proteins, as it has the same effect on BI3K proteins. However, if you compare the sequence of the RBD domain between the species for the different proteins of RAF, it does show a bit of higher similarity than the RBD of B110 proteins. And again, the RAF RBD has similar structure to the same RBD of B110 gamma, although the sequence is different, and it still uses the same interface to interact with RAS proteins. And it's also, again, switch one is the main one that is involved within that the interaction. And if we compare the three interactions together, these are two published data in the previously. This is B110 gamma that I showed before, and this is the RAF RBD with RAS, and this is a model that I built because there isn't a B110 alpha RBD with domain with RAS, so I modeled the RBD of the B110 just to see if there is the similar interface, and I can see that three of them have a similar interface in the interaction. Obviously, there isn't a high sequence similarity between RAF proteins and B110s, but if you look at the structure similarity, there is more conserved structure similarity than the sequence similarity, and you can all see here that the beta sheet that is involved in the action is kind of highly reserved among the RBD domains. Okay, and that's one of my kind of favorite papers, although it's back in 2000, but it's really very nicely illustrating how the switch 1 and switch 2 of RAS proteins are involved with almost every single pathway that they actually are involved in. So basically, none of these interactions can interact with RAS far from either the switch 1 and the switch 2. So there's have been already efforts, obviously, before in trying to find inhibitors for RAS. It's been a big struggle. The most successful one that came so far was Chokatz in back 2013, and he used a specific kind of target of a mutation of RAS, which is G12C, and he used the fact that the cysteine has a sulfur, and he used a certain method and the nucleophilicity of the cysteine, and they applied a certain technique which is called tethering, where they can actually use different inhibitors with sulfate, and then they kind of see which one interacts. And it was actually quite successful and showed a huge success so far. And last thing I know that I actually taking this drug to clinical trials, and it was so specific to the it was so specific to the G12 oncogenic version of KRAS, and basically what it have done, it has actually interacted with the cysteine at around the switch 2, and it blocked the switch 2 from folding in and interacting with GTB instead of GDB, which made this RAS at this case favoring more GDB than GTB. So basically you are encouraging the unactive version of RAS than the oncogenic version of RAS. So, and it showed really incredible success so far. Other efforts, which is now we're moving towards what we're interested in, but it's cyclic peptides, and that's a group in America that constantly use cyclic peptides. They build it on solid phase, and the thing is that the cyclic peptides they build are quite big, but also they are not really very specific, and the size of solid phase, you are limited to the number of peptides you can actually generate. So you're actually talking about thousands instead of more than that. But regardless, the research they do is really very interesting, and they actually faced one of the problems that they do overcome one of the problems that cyclic peptides has, which it doesn't really it has a problem with entering the cell or the cell permeability. So what they have done here, they've done something which is called a double or a bicyclic peptides, which one side has arginine, which serves as a cell penetrating peptide, and the other side has the side of the peptide where it actually interrupts the interaction. So they used it as a method to deliver to the cell, and they showed some really nice success here. But we are interested in a technique that started to come out in the market back in 2000, and it was involved with Ali's when he was doing his postdoc in the States, and it's called cyclic split intine mediated circular ligation of peptide and proteins. And one of the biggest things that came from Ali's lab later, when he became a group leader, he managed to find a cyclic peptide that inhibits the HIF heterodimerization. And it's really quite a nice paper, it showed a successful inhibition of the heterodimer. And then I would like to explain why are we interested, what's the advantage of cyclic peptides above other inhibitors? Well, basically, we are looking at something between 6 and 10 amino acids, so it's relatively good size. The interaction of protein-protein interaction is kind of flat surface. So in order to find a small molecule, the small molecules inhibitors wouldn't really cover this side of surface. So small molecules inhibitors are good in kind of filling up a certain bucket to inhibit interactions, whereas cyclic peptides are big enough to inhibit such flat surface. The C-terminal, the fact that the C-terminal and the N-terminal are linked, it gives it much more rigidity and stability, and obviously it will be much more stable in the cell and will overcome any proteolytic activities in the cell. But advantages of using cyclic peptides or cyclic peptides coming from cyclops, which is a technique I'm going to explain more in details now. It's obviously instead of producing 1,000 as a solid phase, you can actually produce up to 6 million, 10 million, 15 million, and I'll explain to you how we do that. And then this is combined with a reversed hybrid system of screening. So obviously you increase the number of peptides you can get, but then you need something with a good technique where you can actually screen these millions of cyclic peptides. So the split intine was related back to us a very interesting discovery that there's a polymerase that were found in this type of bacteria, and they were found that this polymerase has an N-terminus and a C-terminus, and through a certain expression, they found that the N-terminus and the C-terminus, they interact together and they build a full-length polymerase. And when I took these genes and I put it in E. coli, they found similar they found that the polymerase was also active. So basically, these two genes work in bacteria. Later on, Stephen Binkovic's lab, this is where Ali did his postdoc, they took it a step further. So they tried to use it to see if it can actually cyclize any protein, and they managed to cyclize the DHFR, and they showed that there is the O here represents the cyclic protein, and they also managed to create the first cyclic peptide which inhibits the B0-sterlin F. They came with a certain model of how it works. So this is the cyclop vector, where you have the IC and the C terminus and the N terminus of the polymerase. What will happen is that if you put a target protein in, the IC, the N terminus, the C terminus will fold together, and it will cause an active intine, and the cysteine will of the sulfur will start to interact with the other molecule, which will give you the lariant, and from the lariant you will get the lactone, and the lactone, it will give you a cyclic peptide. So this is the chemistry that they kind of predicted what is actually going on, and having cysteine or a nucleophile amino acid is crucial to initiate this level of cyclization process. So how do they do it? You get the cyclic vector, and you add your peptide of interest, and then you choose, you build your forward primer, and you add a number of random amino acids or random nucleotides that will obviously translate to random amino acids, but you always have to remember that you have cysteine in both sides. You have your reverse primer, and then you will end up with a cassette that will have your protein of interest or peptide, and it will be followed by the N terminus and the C terminus. You do another PCR just to make sure that everything is in frame, and then what you do is that you ligate this cassette back into the cyclop vector, and then you will end up with a tube full of all the combinations that you can get from 6 or 7 codons, and you end up usually with millions, and you'll have a tube full of vectors. Each vector will have a different sequence of a different peptide. So now if you want to develop the screening system, now we have the cyclops, so we need to develop the screening system, and that was used based on another heterodimerization that they found that there are two proteins, B434 and B22, that are they're kind of there are two repressors, and each repressor detects a certain motif or a certain operator in bacteria. The two proteins are very similar in their structure. They only use the 3 alpha helix to detect the operator, and they found that when they managed to get the 434 and just change the codes, the recognition code in the 3rd alpha helix, they managed to get the same dimer that can inhibit the two promoters. And by this you get more strong inhibition, and you have a chimeric protein. Although it's obviously the whole core of this one is still 434, but we still refer to it as B22 just to make it a bit simpler. But we do know that they are both identical proteins. They're only different on the 3rd alpha helix that detects the detection sequence. So we managed to, so this was done to kind of increase the inhibition of two operators than just one. So basically what they have done, that they put this these genes, proteins in chromosomal bacteria with different markers. And what would you expect in this when you have the bacteria in the chromosome? You have these markers that if you transform with the transit vector and you add IBTG, you will end up with constructs like this. So if there is any interaction between the two proteins, what you will get basically a repression, and you have no transcription. And that's the whole basics of the technique so far. The only way you can actually obviously disrupt this and you get expression on selective media is you find cyclic peptides, and the cyclic peptides you can get is that there will be random peptides against RAS, or cyclic peptides against RBD, or cyclic peptides against both. And this is just one of the preliminary results that they have shown with the RBD of B110 and KRAS. And they found that when they growed when they had this construct and they grow in selective media, they did get a shutdown, meaning that there is an interaction between these two in bacteria. But that's just a transit system. You need to build a more stable system with endogenous expression. So all you need to do is you need to take off this cassette, and you go to another vector which contains the R6 origin of replication sequence that it requires a bare type of bare bacteria, and it also have a phage attachment site that will obviously will interact or will overlay with a bacterial attachment site, and you will get an integration. So you will end up with a bacteria that has your cassette and has the same, you end up with a bacteria that has this, your cassette, and these bacteria will be the SNS bacteria that already have these proteins on and are selective on their chromosome. So you will basically, you will end up with a system like this one. So, and this is just going to be your bacteria and your system. Then you will start to use, you will get back your cyclic peptides that you developed, and you will transform them. And this is what you should expect to get. You will grow them on selective media that has all these markers, H3, CAN, and LAGZ. And then you can also load them with a control where it has only the two repressors, so you can try to make sure that you're not getting negative positives or misleading results. And obviously you'll check these results, you check these bacteria, you collect these colonies, and each colony will represent usually one cyclic peptide, and you take it further for sequencing. So what you do after that, you start to choose the best. Am I running out of time?
No, you have 5 more minutes.
Okay, cool. That's more than I need. So what you do is that you build these bacteria or the cyclic peptides that you selected from out of 60 million. You can take them down to 100. You can build them on solid phase, and you can then try to check these if their interaction in vitro by using thermal shift assays, and you can identify and you get more information of the interaction and the affinity of interaction using more sophisticated techniques or obviously biophysical techniques like the isothermal titration and the microscale thermophoresis. And then you can start to work on the improvement, because most of these cyclic peptides, their interaction are usually around micromolar. So this is one of the examples that they used before, my supervisor and Ali from Southampton, that they found a peptide that started with 17 micromolar. With some alanine scanning and some modifications, they managed to increase the affinity. They went all the way down to 18 nanomolar. There are other ways you can also use, instead of alanine scanning, which is peptide array, instead of changing every single amino acid just to alanine, you can actually load it in 96 membranes, and you can change each amino acid to the 20 amino acid to see which amino acid is important that actually can increase the interaction and which amino acid can actually eliminate the interaction. And obviously, we're going to use X-ray crystallography to try to identify the pocket. So the project we're actually willing to do to build these systems in all the RAS mutations to study against B110 alpha and RAF RBD. I will just run through this very quickly. We started the situation with a bit of a problem that the two proteins are repressors that interact 434 and B22 N-terminus, C-terminus, and whereas the interaction with the RBD and RAS are the opposite. So if we follow this analogy where they were constructed on the vector, we will end up with a situation where the B22 and the 434 are far away from each other, and then you can actually have a non-functional system. So what I have done, I tried to maneuver, and I changed the orientation of each of the 434 and the RBD, and I managed to get back to the system where every protein can be close to each other. And I've done that the toxicity test, where you check if there is proteins are toxic in bacteria or not, with under certain concentrations of IPTG, and the proteins that you can see that were not toxic all the way up to 100 micromolar IPTG. And I've done the tests to show that that my system actually works in bacteria with a full length B434, and you can see that I already started to get shut down at a certain increasing amount of IPTG. And I've also done it on a truncated version where I took certain deletions of the B22 and the 434, and I got the same result. So basically, in conclusion or in summary, we are attempting to inhibit the interaction between B110 and RAS and RAF, RBD, RAF, and RAS. We chose cyclic peptides for the size of libraries and their efficiency and the efficiency in inhibiting. And we chose obviously Cyclops, again, that we can get actually basically millions, and we can test them. And we're going to use all this MST and ITC and crystallography to improve it. And at the end, I would like to thank my lab and my supervisor and our collaborator in Southampton University.
induction of the innate immune responses to some of these peptides, or induction of T-cell responses, because cyclic peptides, when they get broken down, present just as well. And you said this is 6 to 9 amino acids. It can certainly be picked up by the MHC class 1. So it would be prudent to do some screening with those before you start manipulating them all the way.
Yeah, so the thing is that it's a good point, but the way we start the technique, you need to find them first. So we'll have to build the cyclic peptides first. And obviously we will, if I get an interesting hit, which is around like 30 micromolar, that's already quite weak to even use it in tissue culture. So I will still, in any case, I will have to use to increase the affinity to get to a reasonable KD or affinity, like I can handle 1 micromolar, I think. And then you will start to see if it has any immune response or not. But the thing is that we deal with one problem at a time. So first you find the cyclic peptide. Usually one of the problems you get is solubility, so you'll have to deal with this problem when it arises. And then you start to use it in cells. I'm not sure the level of immune response you can just get from cell culture, so you'll have to, it would be better if you get it onto mice and see if there's any immune response. I think it's...
I would use human PBMCs.
Yeah, we can start from there. But before this stage, I think it would be better to have a reasonable, efficient, interacting molecule. And this is why we use bacteria to try to just eliminate it, to make it, it is in vivo in bacteria, but it's still in vitro compared to where the genes are coming from.
So my question is also related to that. So do you have any in vivo studies so far?
Not so far. The problem is that as soon as it reaches this level, like the drug that Ali found and published, it was very good. It's working quite well, but it lacked a bit of solubility. Once you reach an interesting peptide, it's soluble, it's effective, they don't publish it, and they make companies. So I'm not really sure. He said this to me, that he said it's already making a company out of it. So I'm sure he've done all the tests. I'm sure all these questions has been asked. But it's from scratch, like from where I'm starting, all the way to start to check in human T cells and to see the response. It's quite a long way. So it's we need to first just identify the molecule. You build your library, identify the molecule. You make sure it's specific for these proteins, increase the interaction, and then you can go down the road and see what they do in cells.
I mean, not in cells, yes, but in vivo, I mean, animals.
In animals, yeah, that's even another, because then you will have to work on a delivery system. So you'll have to, this is where it will start to get to, it's going to be a bit more pharmacological, so we will need a pharmaceutical company to come and to help us with this. So as soon as we want to take it to animals, we will have to see which is the best delivery, which is going to be like lympholyzed or solution, or so these are all things that depends on what the company, or what the suitability of the drug itself.
But I guess if you want to attract the companies, you need to first show some sort of preliminary data.
I think they get interested already if you manage to show, from the examples I know, that if the compound is very specific to the target protein, if it interacts with a reasonable strong KD, and I mean something around 10 nanomolar, and it's soluble enough, it gives good results in cells, and all you need to do is just you need a push to take it to mouse and then see which is the best way to deliver it. Then they start to be interested. I have a long wait before I try to get their attention.
Thank you. Any other questions? Thank you so much.
Thank you.
So